The TANBead 61C reagent is designed for cfDNA extraction from serum and plasma. Let us now demonstrate the complete workflow. First, centrifuge the whole blood sample at 1600 times G for 10 minutes to separate the serum or plasma. Carefully collect the upper layer of the sample without disturbing the lower fraction. Next, centrifuge the sample again at 16,000 times G for 10 minutes to further remove impurities. Transfer 500 microliters of the supernatant into a new conical tube for the following steps. Add 30 microliters of 20% SDS and 20 microliters of proteinase K to the sample. Mix the sample thoroughly, then incubate at 60 degrees Celsius for 20 minutes to facilitate sample processing. Transfer the processed sample into the autoplate, preparing for the extraction process. Insert the spin tips into the Switch 8 instrument and start the tip insertion program. Finally, place the autoplate into the Switch 8 and start the corresponding program to complete the process. Supported by the automated platform, the 61C reagent ensures stable and consistent nucleic acid extraction, enhancing the reliability of your testing and research.